Welcome, everyone, to this lecture on the fundamentals of genetics. Today, we are going to dive into a fascinating and crucial topic known as the coefficient of coincidence. If you have ever wondered how geneticists accurately map the distance between genes, or why certain genetic traits do not always inherit exactly as basic probability predicts, you are in the right place. We will build this concept from the ground up, starting with a simple physical analogy, moving through the mathematical formulas, and finally solving real-world genetic problems together. Let us get started. To truly understand what is happening at the microscopic level, let us start with a physical analogy. Imagine you are trying to tie knots in a very thick, stiff piece of rope. If you manage to tie one knot, the physical stiffness of the rope makes it incredibly difficult to tie a second knot right next to the first one. You have to move a certain distance down the rope before it becomes flexible enough to allow another knot. In genetics, chromosomes behave a lot like this stiff piece of rope during cell division. When chromosomes swap pieces with each other, a process called crossing over, they form a physical twist called a chiasma. Just like the knots in the rope, one crossover physically interferes with the formation of a second crossover right next to it. This is why we cannot assume multiple crossovers happen independently. Now that we have our stiff rope analogy in mind, let us look at how this actually appears with chromosomes. On your screen, you see two scenarios. In the top diagram, we have a high interference zone. Notice the first crossover, or chiasma, creates a physical constraint. The region immediately next to it is the physical interference zone, where the chromosome is simply too stiff to bend and cross over again. In contrast, the bottom diagram shows a low interference zone. Here, the distance between the 2 points is large enough that the chromosome regains its flexibility, allowing a second crossover to occur. This diagram beautifully illustrates that DNA is a physical structure with mechanical limits, which directly impacts the genetic outcomes we observe. Having visualized the physical constraints of chromosomes, we can now formally define our topic. The coefficient of coincidence is a mathematical metric used in genetic mapping to quantify this exact physical interference between crossover events. Formally, it is defined as the ratio of the actual number of double crossovers we observe in an experiment to the number of double crossovers we would mathematically expect if the two events were completely independent. It gives us a precise numerical value. If the coefficient is exactly 1, it means crossovers happen independently, without any physical interference at all. Conversely, a value of 0 means the first crossover completely prevented the second one from happening. It bridges the gap between theoretical probability and biological reality. You might be wondering why we need to calculate this coefficient at all. Understanding the coefficient of coincidence is absolutely critical for creating accurate genetic maps of chromosomes. When geneticists map the locations of genes, they use the frequency of crossovers to estimate the physical distance between those genes. If we ignore the fact that one crossover suppresses another, our mathematical predictions for double crossovers will be completely wrong. This would lead to highly inaccurate gene maps. By calculating this coefficient, scientists can correct their map distances and better understand the structural constraints of DNA during meiosis. Without it, tracking inherited diseases or breeding better agricultural crops would be nearly impossible. Before we can calculate our main coefficient, we must first figure out what we mathematically expect to happen. This formula calculates the expected number of double crossovers by treating the two single crossovers as independent probability events. We use the product rule of probability. The term P1 is the decimal probability of a single crossover happening in the first region. P2 is the probability for the second adjacent region. We multiply these two independent probabilities together to get the combined probability of both events happening simultaneously. Finally, we multiply by n, which represents the total number of offspring observed in the entire experiment. If the map distance increases, the probabilities increase, which physically means we expect to see more double crossovers. Now that we know how to find the expected double crossovers, we can introduce the main formula for the coefficient of coincidence. This formula is a simple ratio that compares what we actually see in the laboratory to what basic probability tells us we should see. The variable c represents the coefficient of coincidence, which is a dimensionless decimal number, usually falling between 0 and 1. The numerator, O, is the actual number of double crossover offspring counted in your genetic cross experiment. The denominator, E, is the theoretical number we just learned how to calculate. If the observed number matches the expected number, the fraction equals 1, meaning zero interference. As physical constraints block crossovers, the observed number drops, and the value of C decreases toward 0. Once we have the coefficient of coincidence, we can easily calculate the actual interference. This formula introduces the concept of interference, represented by the symbol I. While the coefficient of coincidence tells us the proportion of expected double crossovers that actually occurred, interference tells us the proportion that were physically prevented or blocked. It is a direct inverse relationship. If C is 0.2, it means only 20% of the expected double crossovers happened. Consequently, I would be 1 minus 0.2, which equals 0.8. This means 80% of the expected double crossovers were blocked by physical interference. As the physical stiffness of the chromosome increases, interference goes up and coincidence goes down. Let us put these formulas into practice with a concrete example. Suppose a geneticist performs a 3-point test cross with a total of 1,000 offspring. The genetic map shows the distance between gene A and gene B is 10 map units, and between gene B and gene C is 20 map units. Our first goal is to find the expected number of double crossovers. We start by converting the map distances into crossover probabilities, remembering that one map unit equals a 1% recombination frequency, or 0.01 in decimal form. So 10 map units becomes 0.10, and 20 map units becomes 0.20. We multiply these probabilities together and then multiply by the 1,000 offspring. This gives us a final expected count of 20 double crossover individuals, assuming no physical interference. Continuing with our example, the geneticist counts the offspring and observes exactly 5 double crossover individuals in the lab. We need to calculate the coefficient of coincidence and the interference. We take the actual observed number, which is 5, and divide it by our calculated expected number, which is 20. This division yields a coefficient of coincidence of 0.25. This means only 25% of the doll crossovers we mathematically expected actually took place. Next, to find the interference, we subtract our coefficient of coincidence from 1. The result is 0.75. Physically, this tells us that 75% of the expected double crossovers were blocked from happening because the genes are relatively close together, making the DNA too stiff to bend twice. Let us look at a different scenario where we work backward. In an experiment with fruit flies, the map distances are 15 and 10 map units. Previous studies show the interference value is 0.40. If we breed 2,000 flies, how many double crossovers should we actually expect to see? First, we use the given interference to find the coefficient of coincidence by subtracting 0.40 from 1, giving us 0.60. Next, we calculate the theoretical expected double crossovers by multiplying the decimal probabilities, 0.15 and 0.10, by the 2,000 flies. This gives an expectation of 30. Finally, we multiply this theoretical expectation of 30 by our coefficient of coincidence, 0.60. The result is 18, meaning the researcher will realistically observe exactly 18 double crossover flies. We have covered a lot of ground today, so let us summarize the key takeaways. First, always remember the stiff rope analogy. Chromosomes have physical mechanical limits, and one crossover physically interferes with another nearby. Second, the coefficient of coincidence is the vital bridge between abstract mathematical probability and physical biological reality, allowing us to compare observed double crossovers to expected ones. Third, interference is simply the inverse of coincidence, measuring exactly what proportion of crossovers were blocked. Finally, these calculations are not just theoretical. They are absolutely essential for geneticists to correct map distances and build accurate genetic maps. You now have a solid understanding of how physical constraints shape genetic inheritance. Great job today.